genes, you have to have translation of those Cas genes, genes that cut the incoming DNA, genes that help for recombination, Cas9 genes. All seems very complicated, right? But when we go to put it in eukaryotes, to get it in eukaryotes, to get it work in eukaryotes, does it work in eukaryotes? It's really very straightforward. All we have to put in is the gene for the Cas9 and the gene for the guide RNA that's going to direct or target the Cas9 to the location we want to cut. That's all we got to do. We can do more, and I'll show you more, but that's the basics. You just got to get two things in. Now I'm going to show you a somewhat more advanced method, and that's the one we're going to do, because we never want to take the easy way out, right? In our method, we're going to put in three things. We're going to put in a third thing. But those are the two basics. And if you have those two basics on your plasmid, and you can get that into your eukaryotic cells, you too can do CRISPR. Okay. So that's just my little review there. And then we went to this diagram to show you that if you get those two things into your eukaryotic cells, you've got your single guide RNA bound in the Cas9. The single guide RNA has the 20 nucleotides you put in that targets whatever gene you want to target. It'll go to that gene and cut, and now you're halfway there. Well, you're more than halfway. So what we've done so far is to just cut our gene of interest. But other than cutting it, we haven't really done any editing yet, right? So how do we do the editing? Because this is a gene editing technique, right? Yes?
Yeah, I have a question, Steve. The sgRNA is part of the plasmid.
The sgRNA is part of the plasmid, except for the 20 nucleotides that we put in to target our gene of interest.
How do you guarantee that after transcription they are not going to be translated?
Because the guide RNA sequence does not have the signals required for translation. So translation requires signals for initiating translation, and the guide RNAs and RNAs in general that don't get translated just don't have those signals.
So the insert addition is also in RNA language.
Let me just go back to here. So when we put in that 20 nucleotides, that's a DNA sequence. But when it gets transcribed inside the cell, now it's the single guide RNA. So the single guide RNA is transcribed from the plasmid, just like the messenger RNA is transcribed from the Cas9. And this messenger RNA will get translated because it has the translation signal. That RNA will not get translated because it does not have the translation signal. Okay? Yeah.
So it's not an mRNA, it's its own kind of RNA.
Yes, it's its own kind of RNA, but remember there are millions of those.
Right.
There are ribosomal RNAs, transfer RNAs, and microRNAs, and snRNAs, and all these other things that we haven't talked about yet a little bit. Okay? Okay. So there's nothing unusual in cells about having a lot of RNA that doesn't get translated. In fact, most RNA in cells doesn't get translated. 95 to 97% of it is RNA that doesn't get translated. Only 3 to 5% of your RNA in a cell is messenger RNA and actually gets translated. Right? So you can think of messenger RNA as the weird ones. They're the ones that get translated, right? Question or just question? Question. Okay. All right. So we have that now. When we put that in, we get this. We have the guide RNA gets transcribed off the sgRNA gene that I showed you. The Cas9 gets transcribed and translated from the Cas9 gene, and now the CRISPR can happen. And the first step in the CRISPR is the cutting of the target, right? The cutting of the target DNA. Now, what happens after that? Well, what happens after that depends on your goals. And so, first, let me preface this by saying that a double-stranded cut in the DNA of a eukaryote is the worst DNA damage that cell will ever see. And cells have evolved mechanisms for repairing double-stranded breaks because double-stranded breaks are the worst. And the reason that they're worse is if you get a double-stranded break in a eukaryotic chromosome, that chromosome comes apart, and that chromosome is going to get degraded eventually, and that's the end of the cell. That cell's going to die. Okay? So a double-stranded break is really bad, and because of that, our cells have developed multiple repair systems for repairing double-stranded breaks. Why do our cells have to worry about double-stranded breaks? Well, radiation can cause double-stranded breaks. There are chemicals that can cause double-stranded breaks. So cells, all eukaryotic cells and bacterial cells and archaea have all developed methods for dealing with double-stranded breaks. So once that cut is made, the cell is going to try to fix it. It needs to. And so how does that happen? Well, in this diagram, again, we have our guide RNA up there. We would call it, in this setting, we're going to call that our single guide RNA. And the orange blob is our Cas9. And then here's our target DNA, our chromosome that has been, that's had a double-stranded break caused by the Cas9. So the Cas9 has caused this double-stranded break. Now, what can cells do about it? Well, there are two pathways for resolving double-stranded breaks, for fixing them. And the first one I'm going to tell you about is this one, and this is called non-homologous end joining. Okay? If you had your choice, this is the one you don't want. Okay? And here's why. This used to be called an SOS repair system. It's a last-ditch repair system for the cell to fix what's going to be catastrophic damage. Right? And all that happens is the cell has enzymes that say, oh my God, we've got a double-stranded break, we're just going to glue these things back together any way we can. And in that process, without going through the molecular details, nucleotides from the ends can be lost. The cell also tries to add nucleotides back, but it's in an error-prone method, and so you end up with a mutation here. You end up either with extra nucleotides or missing nucleotides. Now, we do this in mosquitoes, and typically what we see with mosquito chromosomes is that we have deletions when there's non-homologous end joining. Okay? So, a last-ditch effort to repair things, but it succeeds in putting the chromosome back together, but if this is in the middle of the gene, you're going to have a mutation. Now remember, most of the eukaryotic chromosome is not gene, and so if this happens in a non-gene region, no big deal. The cell has fixed it, and now the cell's ready to go, right? But if we target a gene, and that's usually what we're doing, then this mutation is going to occur in our gene of interest. Now, why might we want to do that? Well, if we want to mutate a gene to see what the effect of that mutation is on those cells, so a knockout, thank you so much for the recipe. Thank you. Sent out an SOS for some emergency local repair. Okay, so you get a gene knockout this way really easily, knock out the function, and see what the result of that is. There are some experiments where you want to knock out a gene, because if you knock out that gene, you might cause the organism to do something different than it does normally that you would find desirable. So for example, just from our own work, we have been working on knocking out genes that are involved in sex determination in mosquitoes, so that if we mutate the right genes this way, we can get all female mosquitoes, and then that's a problem for the mosquitoes, right? So that's just an example of what you might do. Now, in many cases, and I'll even say in maybe half or more than half of the cases, this is not what you want to have happen. Often what you want to have happen is to make the cut, but then replace that with some other DNA. And so in this case, we'd be doing, instead of a knockout, this is what's known as a knock-in. You're going to knock in a sequence using CRISPR, okay? So we're going to add an additional sequence. Now how does this work? This is a different repair system. This one's non-homologous end joining. and HEJ. This one is called homology-directed repair. And this is the one that can be used to fix damaged genes or to change genes so that it gains a new function, right? So, so let's take a look at this. And this diagram is not the best, and I'll redraw it if you want me to. So, this region here on the chromosome and this region here on the chromosome, on our donor DNA, we designed the donor so that this region on the donor matches this region, and this region on the donor matches that region. And I think the artist screwed up here because the longer one should be on the side and the shorter one on the side. But anyway, it's okay. This sequence matches that one, this sequence matches that one. Don't worry about the length, they're a match, okay? And typically we design our donors so that 50 nucleotides match on this side of the break and 50 nucleotides match on that side of the break. And we're actually going to do this in our experiment. Then in the middle, you have the sequence that you want to insert into the genome. Again, this could be adding to a gene coding for a protein. It could be adding a new domain to that protein because you want to do a protein modification. Maybe you want to, for an industrial process or for a vaccine, you want to put in a new kind of domain in the protein for functionality. Okay, so we design this donor. Now, if you design the donor, how do you make it? And the answer is that you make the donor just by synthesis, and you can order this from a company. And in fact, the donor that you're going to use in this class is going to be one that that we synthesize. It's about 150 base pairs. It's got 50 base pair match to the chromosome on this side, 50 base pair match to the chromosome on this side, and then about another 50 or 60 that we're knocking in. Okay? Now, this is the third thing that we put in. When I said we put three things in, this is the third thing. And so the third thing is this donor DNA. And the donor DNA is what you put in if you want to make a specific modification to the chromosome here. And I'm going to show you an example from human medicine using sickle cell anemia as an example, where this can actually be used to help cure a human disease. Has been used to cure a human disease. Okay. So everybody clear about this? You put in the donor DNA. The donor DNA, just like the plasmid, finds its way to the nucleus, and all these pieces find one another, and you get the break, and then this recombination event can occur. This is called homology-directed repair because now the cell, instead of just slamming these two sequences together willy-nilly, is fixing this break by doing this recombination. Now, how does that fix the break? Well, if you track along the chromosome, you go along here, you cross down, you pick up the sequence, you cross back up, and you keep going, and now your product of this recombination event is an intact chromosome. Okay? So again, the chromosome would be going out this way in both directions. We track along, we cross down, we pick up this sequence, we cross back up, and we have an intact chromosome. We also generate a small piece that's got a break in it, but that just flows away and gets degraded. And then we end up with this that has our new sequence knocked in. That new sequence, as I said, could be a new domain in a protein, or it could be replacing a sequence here with a modified sequence that repairs a mutated gene, like in sickle cell anemia. Yes, sir?
Is there a limit to the length of the amount of insert in that donor DNA?
Don't know what the biggest piece that's been put in, but if you want to put in really big pieces, there's another way you can do this where you put in two different passes that cut the chromosome in two locations far apart, and then you knock out that big sequence and you can replace it with another big sequence. That's lower efficiency to do that, but it works. In the class, what we're going to do is we're going to do a break with our Cas9 enzyme in the E cells in the adenine 2 gene, and then we're gonna knock in a sequence. And after we're nearing the end of the experiment, what we're gonna do is we're gonna sequence this region of the chromosome in yeast of the adenine 2 gene, and we're gonna see if the repair occurred the way we predicted, and did we knock in the sequence that we were trying to, okay? It's a pretty cool experiment. So we're not doing the easy one, we're doing the hard one, to make it more fun, okay? Any questions about the two repair pathways? Oh, I know what I need to tell you. So in real cells, how does this work, right? In real cells, you don't get supplied, if one of your chromosomes gets broken, you don't get supplied with a donor DNA to fix it, right? So in real cells, it's a bit complicated, but if this break occurs in your chromosome 1, you have another chromosome 1, you have the homolog, this is one of the advantages of being diploid. You have another chromosome 1 that can serve as the donor. And so what happens in the repair system in cells is that the other chromosome serves as the donor, and there's a replication event that occurs so that the donor chromosome doesn't lose the piece. So the mechanism involves a replication event so that the donor repairs the broken chromosome, but the donor itself also stays intact. Wouldn't do much good to fix one homolog and then damage the other homolog in the process, right? So in your cells, if you get a double-stranded break, this is what you hope happens, is that you repair your chromosome with the other chromosome, with the homolog, right? This is going to be error-prone, this is not error-prone.
What's the signaling? How does the other know that one is broken?
When there's a break in a chromosome, there are enzymes that monitor for breaks in the chromosome, and when there's a break in a chromosome, there is a signaling pathway that tells the cells to really crank up the enzymes that are involved in this kind of repair. And same thing with this pathway. If there's a break, the enzymes involved in this pathway get cranked up too. So if you experimentally introduce double-stranded breaks with X-rays, let's say, into cells in culture, and then look at the expression of their repair enzymes, those repair enzymes crank up in response to the damage. Yes?
If you're trying to repair a defective gene, wouldn't the chromosome, the other chromosome also have that same issue when you're trying to fix it, when donating DNA to fixing the first chromosome?
So if you're talking about somebody with sickle cell anemia, a sickle cell anemia patient has both chromosomes with mutations that's causing the problem. And if you can go in and fix one of those, you've solved the problem. Because a heterozygote person that's got one sickle cell allele and one wild type allele is pretty much okay. They have something called sickle cell trait, but they're mostly fine. In my lab a few years ago, I had sisters, one of whom had sickle cell anemia and the other was heterozygous. And the one that was homozygous for sickle cell anemia had real problems, and the one that was heterozygous rarely had any issues at all. So if you replace, if you can fix one of the two chromosomes, it solves the problem, and I'll tell you a really heartwarming story later on. Any other questions? Does it make sense?
So when you cut the DNA, it's going to cut the DNA on both chromosomes, right?
When you do CRISPR, so CRISPR is not the most efficient process. And if you get those enzymes into the cell, you might CRISPR both chromosomes, but there's a high probability that you would only CRISPR one of the two chromosomes. So in a lot of cases, what you're going to do is, if you're doing this for medicine, you've got an individual who's got a homozygous recessive mutation that is not good. And if you go in and you can fix one of the two chromosomes, then you've solved the problem. Because there are some dominant diseases like Huntington's that you need to fix both. But most people who have Huntington's are heterozygous. Anyway, I digress, we can go back into that later, yeah.
One question, so you always hear like things about off-target effects. When it comes to CRISPR, does that just mean it's just cutting somewhere else?
Yeah.
Not where you want to do
So
it?
when people talk about off-target effects, the off-target damage, the issue is that you've got these 20 nucleotides. We're limited to 20 because that's what Cas9 does, right? If you make this 25, it doesn't work anymore, right? So you can't improve the specificity by making this longer. So you're stuck with 20. Now, 20 is a lot, and you get pretty good specificity with 20. The problem is that with 20, it'll find the right place and it'll hybridize to the right target. But it'll also find other places where maybe it's got an 18 out of 20 mass or a 19 out of 20 mass and hybridize and cut there. And so if that happens, you're getting cutting in places that you don't want. And those are the off targets. Off targets are just targets that are getting cut that you don't want, right? So that happens, and there are improvements in the protocols that have improved specificity in recent years, but it's still something you have to watch out for. So with our mosquito experiments, for example, when we've had our mutations that are on target, we're still not sure if an off-target change occurred somewhere else. And the only way we know that is to sequence the genome. We've got to find if there's another modification that's occurred elsewhere in the genome.
You hear a lot about prime editing as well these days. What is that?
So let me come to that later. That's a more advanced topic. I'll come to that later.
Are both of those repair pathways in eukaryotes and bacteria?
The double-stranded break repair pathway. So I'm not a bacterial geneticist. I know that bacteria have double-stranded repair. I don't know that they have both of those. I just don't know. I can look it up. But this is in eukaryotes. Okay? Yeah.
When they used CRISPR on the three females that was born in China, were mutations introduced to those?
Yeah, so I'm going to come to that. That's in my slide deck here, so I'm coming to that. Okay, so let's go back to here. As we walk through our experiment, since we're doing this homology-directed repair, which is more complicated, we're going to diagram this in detail, and so we're going to look at this a lot more. So don't, if it's not 100% clear to you, we're going to go through it again, so not a problem. Now, as I've said over here on the side, we can use non-homologous end joining to knock out genes to study function. That's genetics, right? Having mutations and studying the effects of mutations is really classical genetics. The difference is now we can introduce mutations anywhere in the genome instead of just introducing mutations randomly. We can repair damage to genes using this homology-directed repair, and that's what we're going to do. We're going to do HDR in our experiment, so we'll go through that in a lot more detail. You can replace portions of genes, as I mentioned, to engineer new functions, or you could even put in an entirely new gene using this method that I was talking about, where you make two cuts and then put in a really big fragment. And the key about all this is you're doing this in live cells. This is not something you're doing, in vitro to DNA in a test tube like a lot of the experiments you're doing. This is in live cells. And so you make these modifications, and if the modifications don't kill the cell, now you've got a cell line that has the modification you want. And that's really powerful. So in humans, this has been done on a variety of somatic cell types, muscle tissue, heart tissue, skin tissue, and so on. A lot of somatic cell types. It's also been done in germ cells and embryos, but the rule was that if you did this kind of modification on embryos, that the embryos would not be implanted and would not result in a human baby. The first work on human embryos was CRISPR on non-viable embryos that were not implanted for pregnancy. But in 2017 in China, the scientists did it with two families. In one of the families, twins were born, and in the other family, one girl, and they all had a CRISPR modification that just created a huge furor. So what did they edit? What they edited was the CCR5 gene in the human genome. The CCR5 gene encodes a receptor on cells that HIV binds to. And so the argument was by editing the CCR5 gene, by knocking out CCR5, that then these baby girls would be resistant to HIV. And that's true. There are natural CCR5 mutations that have been seen in the human population that make people resistant to HIV. The problem is that people viewed this as human enhancement. It wasn't curing a disease, it wasn't curing a problem that these girls were likely to have, and so that's human enhancement, and that's where the fact that it was done on humans and resulted in a live birth really made people angry, but the fact that it was an enhancement made people even more angry, right? So if instead of doing it this way, if it had been done for some disease that it was clear that the girls were going to have, then I think people would have been, they still would have been upset, but they wouldn't have been as upset, I think. It's not just HIV resistance that this imparts, but there's some evidence that by mutating CCR5, in mice at least, the mice are smarter, have faster stroke recovery, but it also might make them more susceptible to Zika. And then another thing that wasn't clear was whether the researcher checked for off-target mutations, and that was probably not done. So whether these girls have off-target mutations or not is not known. I think the Chinese government is doing the right thing in keeping their identities a secret and not talking about it so they can live normal lives. But we don't know the answer to those questions. In this part of the diagram, I'm not going to go through this, but it's kind of a timeline of what happened and how it was discovered what had occurred. The crazy thing is he actually went to a meeting and announced this at a meeting, I believe it was in Hong Kong, and people were just blown away and really unhappy, and he certainly didn't get the reception that he thought that he was going to get. Anyway, China issued new regulations after this for gene editing, outlawing doing editing on embryos, and in December of 2019, he was sentenced to prison for 3 years. He's now out of prison, and if you look online, you can see he's talking some crazy stuff. So, it sounds like he's trying to get himself in trouble again, like he wants to go back to jail. Anyway, yeah. So that is that, and it's never happened again since then, as far as anybody knows, and nobody, at least publicly, has any plans for doing so. Now, the situation with sickle cell anemia is different because this modification was germline modification, so that the babies that were born, every cell in their body was modified, and they're going to pass that to the next generation, right? So that's called germline modification, and that's a big no-no because people see that as messing with, the sort of natural evolution of, humanity, right? But doing it on somatic cells, people feel like that's okay. People were uncomfortable with that at first, but over time, people have become more comfortable with that, and so now the work on sickle cell anemia patients is really celebrated.
Question?
Yeah.
So they all the girls had an HIV positive father, is that right?
Mhm.
But there was no risk to the actual children of the
So children are born from HIV positive fathers all the time without HIV, and the reason is they can take the sperm and they can wash the sperm to get rid of the HIV, and so that was a bogus argument here, really, because there are other ways of doing it that work just fine. Okay, so that was the argument, but people weren't buying it. CRISPR has been done on a wide variety of eukaryotic organisms now. We're doing it on yeast. I don't think I put yeast on the list here, I should. We've done mosquitoes. A lot of people have done a lot of other species. So CRISPR, I mean, the list of species is probably in the hundreds now. So let me tell you about sickle cell anemia. This is, these are the globin genes, and the globin genes are on two clusters this is in humans. There's the alpha globin cluster on chromosome 16, and there's the beta globin cluster on chromosome 11. On chromosome 16, there are 1, 2, 3, 4, 5, 6, 7 alpha globin genes. But they're not all functional. This one, anything that's got the psi in front of it, so that one and that one, those are pseudogenes. A pseudogene is a copy of a gene that's non-functional. So over evolutionary time, what's happened is that the globin genes have expanded. So once upon a time, the idea is that there was one globin gene, and then that globin gene duplicated, and now there were two globin genes. And then over evolutionary time, there was some recombination between chromosomes, and one of those globins moved to another chromosome, and then the two globin genes got duplicated and duplicated again, and now there were two globin gene families. And over evolutionary time, the alpha globins and the beta globins work together to make hemoglobin. So in normal hemoglobin, I believe, it's 2 alphas and 2 betas. Do I have that right? Yeah. Okay. 2 alphas and 2 betas. And the different globins are expressed at different times during development. So early in development, you get expression, so the purple ones are the ones that are functional. Early in development, you get a progression of expression of these genes. And then finally, in adults, you get expression of these down here. Mostly, I believe it's alpha-1 that's expressed at the highest level. In the globin gene family, we've got 1, 2, 3, 4, 5, 6 beta globin genes. Again, we've got a pseudogene here. So again, a pseudogene is a non-functional copy. So during evolutionary time, the gene duplicated, and one of the copies got a mutation and became non-functional and stayed non-functional. So you can tell that it's a globin gene, it's part of the family, but it's non-functional. It has no promoter, and it's got mutations in it that clearly show that it wouldn't be a normal globin. So stop codons in it and so on. Okay? So these pseudogenes are not expressed. They're not transcribed, they're not translated. Again, in the globin gene cluster, these are expressed during development, early embryos, later fetal development, and then finally beta globin is the adult version, the version that we all have being expressed. Sickle cell is caused by a single base mutation in this beta globin gene. I believe it's in the 6th codon, a single nucleotide change that causes a single amino acid change. And that's what causes most sickle cell anemia. So it's just a single base change, and that seems like if you're going to do CRISPR, that would be pretty easy to fix. So that would be one way to fix it. That's not the way that they decided to do it first. This is being done now, but originally what they decided to do is that this delta globin here works just fine in adults. And there are some adults that actually express both for some reason. That's a rare situation. So what they decided to do was, instead of modifying the beta globin gene, because they were concerned, they didn't want to do any further damage, right? So what they decided to do was instead to turn on the delta globin gene. And the way they did that was to do a CRISPR modification in the promoter region that took out a region that inhibits the expression of delta globin in adults, right? So they modified the promoter so that the delta globin gene can be expressed in adults. And they hoped that that would work, right? So when they did this, the broken beta globin gene was still broken, but they activated this delta globin gene. So what happened? Well, here's Victoria Gray, and it's really a great story. So NPR has had an interview with Victoria Gray ever since she became the first patient to be treated with CRISPR. I believe she's from Tennessee, I want to say, but she had really bad sickle cell anemia. Hers got worse as she got older. I think she was in her 30s when she had the treatment. She had 3 kids, really had a hard time just with normal life, and she would be in the hospital every couple of weeks with really severe pain. Quality of life was very low, and she's been really open that she thought about suicide, and the only thing that kept her going was that she's got 3 kids, right? So, so she was a perfect candidate for, an experimental therapy, and volunteered. Her doctor that was treating her for sickle cell happened to be involved in the project, and so she was the first patient. You can follow her story by looking up these interviews with her on NPR, and it's really great because at year 1, she's talking about how she hasn't had a sickle cell crisis in a year, and she's hopeful that it's going to last. And then year 2, still no problem. Year 3, no problem. And now she's out to about year 6, and she has never had a sickle cell crisis since. So even though she's still making non-functional beta globin, she's making enough delta globin that binds with alpha globin to have enough functional hemoglobin that she has no sickle cell crisis anymore. So that's pretty cool. And because of the success with Victoria Gray, this has now been done on a pretty large number of patients. And because it's worked so well this way, now I believe there are going to be clinical trials starting where they're actually modifying the beta globin gene. Yeah.
So does your blood cells still have that shape?
Some of them do. So blood cells that have the sickle cell hemoglobin, they get that sickling shape and they clog capillaries. So they looked at her blood. She does have some of those cells, but she has enough of the normal cells that it's not clogging up her capillaries. It's not causing any of the pain and other symptoms that you get from sickle cell. So obviously she's thrilled, and she's actually become a spokesperson for encouraging medical research and encouraging research using CRISPR to cure other diseases. And she goes to meetings and gives talks now, and it's pretty cool. So again, this is a somatic cell modification because what they're doing is they're taking her bone marrow cells out that have the precursor cells, the stem cells that produce the blood cells that produce the hemoglobin, right? And so they're modifying those cells, and then they're putting them back into her bone marrow. The amazing thing about this is they're not taking out all the bone marrow from all the bones. They're doing it, I think, in just 1 or 2 places, and it's enough that it's cured her. It's really pretty an amazing story. Yeah.
Do you know if it was like just 1 treatment that she received or a series?
That I don't remember. I don't remember if she needed multiple bone marrow transplants or not. I only remember 1, but I have low confidence in my memory on that one. Yeah.
That means all 3 of her children are heterozygous for sickle cell.
Yes, that's right. That's right. Her husband has been tested. He's not heterozygous, or his kids are going to be fine. But she's thrilled because she gets to go to her kids' baseball games and stuff that she could never do before. So I think it's important. Sometimes and of course, doctors, if they can cure disease, so they wanna do it, right? And there are people who, worry about this kind of modification, genetic modification of humans, the slippery slope to, how many of you seen the movie Gattaca, right? So, if you haven't watched Gattaca, it's 25 years old now, but still really prescient, right? Really good movie. Again, the cat's out of the bag, and I think that, putting the genie back in the bottle is not gonna happen now, so this is gonna happen for other diseases. And my way of thinking, I think it's really great. Now, there are concerns about, this could eventually lead to germline modification and so on. I think that this requires, vigilance and the scientific community and the public just being vocal about what they think. Certainly this is a great therapy. Now, the other problem with it is that it's really expensive right now, and so there's worry about that this kind of thing might only be available to the wealthy and that sort of thing. That hasn't happened yet, because of course these clinical trials have been supported by the government, but in the future, who knows? So that's another thing people worry about. But people said the same thing about surgery 100 years ago too. So I think it's great, but everybody has their own opinion.
Most people are self-experimenting.
Oh, I have no idea. That's the thing, CRISPR is pretty easy. If you have the right know-how, you can probably do CRISPR in the garage. It wouldn't be that easy on, mammalian cells, because you have to grow mammalian cells in culture, and that really requires a tissue culture suite and all that. So probably not in the garage, but, people worry about rogue countries, again, like North Korea or something, doing human genome modification, and, I don't know. I'm a simple molecular biologist, not a politician, I don't know what to do about that, but yeah, it's here to stay, and I think what we need to do is just really keep an eye on things and make sure that it gets used properly.
What's the sort of immediate next disease target that people are going after?
There are a lot of rare heart diseases that are caused by mutations in heart cells, and the idea is that they can make modifications to stem cells that can then be grown into heart cells, pluripotent stem cells, and then put back into the patient. So I know that kind of work is going on, but I'm sure for lots of other diseases also. Progress probably is going to be slower in the next 2 years because of funding situations, but yeah, we'll see how it goes. Okay, I need to get you downstairs in 15 minutes. For CRISPR, that's what I have to say before we do the CRISPR experiment. Now, as we're doing the experiment, we're going to go through all the steps, and we'll talk about the detail, and we'll do the sequencing. So it's not the end of CRISPR, it's really just the and